All righty, I'm going to share screens here. Oh, actually, Winnie, can you give me screen share?
Yep, all good.
Okay, can everybody see my slides okay? Great. All right, so as I said, we're going to take the first half to talk about augmentative technology, which will become segment 5 of the roadmap. And then we're going to switch gears in the second half and do a review or check in on where things stand with the research plan. All righty. So maybe talk about some definitions and give some examples. This is not an exhaustive list, but let's first just introduce this idea of augmentative technology. So first off, you might have heard different terms being used that are kind of synonymous. There's some nuances there, but people often refer to this area as augmentative technology, sometimes assistive technology, sometimes augmentative alternative. There's different formulations here, but they all sort of mean something very similar. I think the assistive describes more communication devices, which we'll talk about in a second, and augmentative is sort of more the general category. So maybe to get things really concrete, we can talk about maybe devices that are even in use right now in people's homes that are benefiting 8P heroes, specifically referring to communications devices. And we'll talk about that, for example, tablets and other devices that, again, may be in use right now. And we would love to actually hear some feedback on how families use these devices today and what the limitations are and where there could be areas of improvement. So I'll focus a little bit about that in a sec. And then we'll talk about what are called neural interfaces. Sometimes they're also called brain-computer interfaces, BCI, or brain-machine interfaces. This is a large category. I'm only going to focus the discussion on what are called non-invasive interfaces. Those are in contrast to invasive interfaces. You might have heard of a company called Neuralink, one of Elon Musk's company that's trying to implant a device, a stimulatory device, and a device that can detect brain waves, EEG, essentially, and that is really used in these. That is really contemplating these more severe cases of disability, like complete paralysis. But we're only going to focus on non-invasive interfaces that don't require any surgery or any implantation or anything actually put physically into the brain. And then the third area, which is also a big area, and sometimes people refer to these interchangeably, but we talk about neurostimulation or neuromodulation. And that's where I'll give one example, and then I'll hand over to Actipulse to give another example. So I want to make this a little bit interactive. And as we go through these examples, I would love to have 8P families maybe chime in here to talk about their real-world experiences with these devices. So first off, these augmentative and alternative communication tablets, or AAC tablets. So I don't have a good sense here, but maybe any of the families who are on the call, if you use one of these devices, maybe you can, I don't know, raise your hand or make some kind of indication here on that in Zoom here that you have some experience with this device. And I'll maybe give a second or a minute here for anyone who wants to chime in, any family that wants to chime in on how they use this device. Please do.
I'll go ahead and share. I have a 7-year-old hero. She has inversion, duplication, deletion 8P, and we do use AAC. For communication, we have used it for over 2 years now. First starting very simply, choice making, 2 options presented to her. Actually backtrack, first started low tech pictures, handheld physical pictures. Moving on to a device, an iPad is what we're using. Look, again, minimal options, field of 4, moving our way up. She is very successful at making choices and able to also ask for basic needs. I need water, I need a drink, to go potty, those type of things. And we're now working on more what I'm considering functional language. So in 2-step processes. So it's jumping from one page, you might click I want, and then it jumps to a list of 25 options that she can communicate with. So we're working on that right now. So I've only seen growth. We haven't seen any regression in her skills with this. It's effective. It's just takes time.
And do you have someone else, like a professional who comes in as well? And can you talk about what that's like?
Yeah, so she does. She sees 3 different speech therapists between school and her private therapy programs. And so they're all working on it. There's a lot of coordination on my side as mom, making sure they are all communicating with each other, that we're all on the same page. You know, what we're seeing at home is what they're seeing there. She did kind of go through a phase where she didn't want to work for anybody but mom, but we worked through it. So there is definitely that component of, as a caregiver, you have to be involved, because also she's not going to learn how to use this at an hour and a half of speech each week. So it's also communicating at home and where we can use it at home and implementing it here.
Awesome. Thank you, Katie. Would any other AP families like to talk about their experience with an AAC tablet? Maybe one or two others can chime in. Oh, Faye is putting a comment here. We have never went to an AAC device. It started with disability signing before going to spoken word. Thank you for that. Anyone else want to share their? Oh, go for it.
Yeah, I can. I think this is a big part of our community. And so my daughter also uses currently a program called Touch Chat. You can download it, and it's in the App Store onto your iPad. And so there are about 42 options in terms of words. And then, you know, if she presses the social button, there are things like hello, thank you, goodbye, I'm sorry. And so she can kind of, after many years of occupational therapy, to get her fine motor skills to be able to point and activate the button, and understand that in combination with speech and language therapists and special educators. So I feel like it's definitely a 9 to 5 practicing every day of the week for her, with kind of a team of different specialists that can work on her auditory skills, her ability to recognize what those icons or words mean. And then we kind of combine that with some other, I don't know exactly, Ethan, what bucket it goes into, but we do things with different electrical stimulation devices on top of it. Kind of like, I guess, if people did physical therapy or like a TENS machine, but I think this is a little bit more advanced in terms of the frequencies and the options there. And then some bioresonance equipment that we have that probably wouldn't be, you know, so recognized in the US, but maybe more in the other European countries.
Great, thank you. Anyone else would like to chime in on their experience with AACs? So if not, then maybe we can go on to the next example. Obviously, this is the very probably the lowest barrier to entry in terms of integrating this into your life and sort of it being non-invasive, obviously. We can kind of ratchet up the next level here when we think about these neural interfaces or these brain-computer interfaces. And so the basic concept here is, can a device, essentially an EEG that can pick up brain waves, can that device then communicate directly to a computer, let's say, and for example, project an image based on those brain waves or thoughts or sentences or words, and instead of even connecting, you know, the EEG output to a computer, what if it connected to a robot that could go move around and do something, right? It doesn't really matter what the machine or computer interface is, but there are many form factors also that these kinds of BCIs have taken, or not, these BCIs have taken. So some of these look very futuristic and very Star Trek-y. Some of these have probably been, maybe have been associated with some quackery, or people are, might look askance at some of these. But I think this field is not going away. And I think with AI, this is only going to get more sophisticated. And you can just see examples here of headgear. These are all essentially the sensors that are picking up the brain waves without having to actually breach the skull. And they take many form factors, as I said. So I'm pretty sure that no one in the audience has volunteered or has integrated one of these into their routine yet. If anyone has, then please speak up. But I would imagine that this is probably the next frontier in terms of augmentative therapy beyond the tablets and the communication devices. So there's obviously lots and lots of potential for this technology to grow, but it's going to need to be sandboxed in potentially in different communities. And maybe the AP community could be such a sandbox where some of these devices are, or some of these interfaces are actually deployed. So I guess maybe this is, since no one is probably using these devices, I guess maybe a quick poll of the families. Is this something that would be interesting to you? Would you have to understand more what actual gains or what actual benefits this would produce in day-to-day life before you would maybe volunteer for such a pilot study? But maybe you can take the temperature of the families to understand, what do you think about when you see this and think about what uses this could have in improving the life of your hero? Who wants to chime in? Maybe Vina or Katie or any of the other families want to chime in?
Yeah, I think that, you know, as an adult, there's a lot of things that kind of are in the market. I mean, you can order things on Amazon that just are like TheraBands to reduce stress and anxiety. And so it's awfully confusing for families. And especially now you add on the fact that most of our 8P heroes are kind of the pediatric population that don't necessarily have the cognitive ability to understand to keep something on their head. So I think often, you know, we do therapeutic listening programs, which is just headphones essentially. Sometimes they have bone conduction. But to even keep that on their heads for protocols like 16 and, you know, 2 hours a day is too much. Generally, they don't understand that has to stay on their head for a reason. Some do, for sure. I think there's a challenge. One of the other things that I think would be amazing is like to explore kind of more smaller wearables, you know, just kind of like the way we think of our Apple Watch. That might be, if we thought about some sort of intervention that could help a little easier for, you know, the younger kiddos, I think, that are affected. And yeah, I mean, frankly, I just think that I'd really like to understand, and I'm excited that Actipulse is here to really hear what they're working on.
Yeah, I think this is a great point and that's why I want to do this, is that, you know, people aren't thinking about pediatric form factors, or not thinking about the simple fact that maybe these children will not tolerate or accept that this object on their head, and so how would you design around that feature? And I think giving this kind of feedback to companies that are in this space is key, because it's one thing to serve an adult market, it's entirely another to serve a pediatric market, especially with these concerns that may not occur to an adult or someone designing for adults, let alone children with a disability. Anyone else want to comment on brain and computer interfaces and whether this is something that just seems too sci-fi? You're going to wait till this technology gets maybe smaller, more convenient, more accommodating of children. Anyone else want to share? Any other family?
Faye has something to say.
I was just going to say real quick that Matt's once had a ECG where, you know, they had to soak it in salt water, like, so that it would work, right? And like that was like, she would have actually probably been fine if it hadn't been for the salt, because it was itchy, right? So like every little key thing like that is going to make a difference.
That's a great point.
And yeah, so there's that. And also, you know, when we put her in with reading glasses, I remember thinking, oh, she is absolutely never going to wear this. This is going to be a disaster. We need to like bolt them, right? No, because as soon as she put them on, she could see. So we never had an issue. So I think that's the thing, is if there's benefit for the kiddos, they'll wear them, right? So
great point. No, this is great feedback. Very great feedback. Thank you. Anyone else want to chime in? Otherwise, I'll move on to the next example. Okay, great. So this is, again, a pretty...
Heidi, did you want to say something?
Oh, apologies.
Oh, I was just gonna, I have a 4-year-old, so she's still quite young, but I was gonna kind of reflect on what Bina and Faye both said. We've had difficulty with Baha hearing aids. Maybe it didn't make a difference for my daughter enough for her to keep it on, but she was uninterested. But we've also had great success with a CPAP mask, which is much more invasive, but she probably felt benefit from it. So I think what Bina's saying, these kids who don't understand aren't going to keep it on. But what Faye's saying is, if there was something that they felt benefit from it, I think I just try not to underestimate what my daughter's capable of doing. But I think these are great comments.
Yeah, this is great. Thank you for adding that. Anyone else? I don't want to cut anybody off, but I'll move on to the next slide if not. Okay. So neurostimulation, neuromodulation is, again, a very broad sweeping category. I want to show you one example here. Maybe it seems a little bit science fiction. Let me see if this will, hold on, let me just do a quick stop share and make sure I can get this video teed up. Okay. Okay, so does everyone see this video?
Yes.
So this is a wearable, technically kind of an exoskeleton, as they say, but also provides neurostimulation. And this patient has MS, and you can kind of clearly see before and after, pretty dramatic. So again, this may not be directly applicable to 8P heroes at this current stage in life, or maybe for some never, but this gives you a sense of where this technology is going. everyone gets the idea here. And then with that, I think it's a good chance maybe to segue to Adrian and Gabriel from Actipulse. Very pleased that you were able to join us. And why don't you two gentlemen take it away? Maybe you can present here for about 10 minutes or so about your technology, and then we'll save a little bit of time for Q&A before we jump over to the roadmap review.
Sure. Hi, Ethan. Hello, everyone. It's always nice to see you, Ethan. I'm Adrian. I'm the co-founder and CEO of Actipulse Neuroscience, and I'm here on a call somewhere. It's Gabriel, our co-founder and CSO. So I'm gonna just share my screen real quickly, just show you what we have been doing at Actipulse for the past 7 years. So it's always nice, to resume 7 years of work in a few slides, but that's part of our life nowadays. So at Actipulse, we're specialized in non-invasive brain stimulation for a variety of indications, which I will show you. We're part of the Paris Brain Institute, as you can tell from my accent. I'm actually French, and we met Ethan through Y Combinator, and we're also part of StartX, Stanford's accelerator program. We founded a company in 2017 already. I co-founded with Gabriel, who's here on the call, who's a medical doctor, and he did his PhD in neuroscience. I'm more on the business side of things. We're 31 people in Mexico and in Paris. And what we have been doing, so our goal was, okay, neuromodulation today, it's the most famous ones are either invasive and non-invasive. So invasive, you most probably know deep brain stimulation, mostly used for Parkinson's disease patients, and TMS for transcranial magnetic stimulation, which is non-invasive, uses magnetic fields compared to DBS, which uses electricity. And TMS is mostly known for the treatment of depression, so treatment-resistant depression. It doesn't require surgery, but you need to go every day for 30 days to the hospital, to the clinic to get your daily treatment in order to reach remission. Our goal for Actipulse is, when we met with Gabriel, is that how can we take the best of these two technologies, TMS and DBS, but make it a wearable from home, so that patients wouldn't require surgery and wouldn't require to go every day to a clinic to get treatment. Because even though TMS works quite well for patients with depression, the biggest challenge is that patients need to go every day to a clinic for 30 days, which limits a lot the treatment and the democratization of this technology. So we created a platform, therapy approach. In neuromodulation, you can modify certain aspects. We can modify the pulse waveform, the pulse intensity, the pulse frequency, the coil form and the location of the coil, and obviously the dosification. So with this approach, we know that we can treat different, we can target different biological effects and different mechanisms, which are shared among diverse CNS indications. And this we have proven with some studies and with some literature review. The ones on the blue side is that we're using high frequency and low intensity magnetic pulses. By targeting these mechanisms, we could potentially target these indications. So obviously we can treat everything. As a company, we need to choose. And this is our clinical pipeline, which we've been doing for the past 7 years. So our most advanced one is for the treatment of major depressive disorder. So depression, to be exact, is treatment-resistant depression. So patients that don't respond to medication. And right now we're currently in our phase 3 study. So patients take the device at home and they use it twice per day for 6 weeks in order to reach, hopefully, remission from the comfort of the home. Our goal here is that patients who, at first, we need to, because of the regulation, we can only use it for treatment-resistant depression. But our goal later down the road will be that patient can use it, as a first-stage treatment, so that they don't require any type of. They can either choose, neuromodulation or medication, or to be realistic, it would be to both using at the same time. That's what our end goal, to be exact in the future. And two things that we've been using lately is for blood-brain barrier permeability for drug delivery, and glioblastoma, which is a brain tumor. So this one, blood-brain barrier permeability, is very interesting because it opens the fields to treat many types of disorders that require drug delivery, but which obviously the BBB is making it difficult to target, right? So we call it pharmatech. So how can we use neuromodulation with drugs so that patient, we can increase drug biodisponibility and increase drug delivery to the CNS. So we have in BBB, we already had one study, actually in a preclinical study, which we saw that we actually can go through the BBB, which we're very excited. By the end of this year, we will have around 60 mice to prove our technology, and we have divided this in 3 targets, right? So first are medication, so drugs that really cannot go through the BBB. So that's all the drugs that are stuck in this clinical study, hell that cannot go to phase 3 or cannot go to their phase to market approval. And the other type of drugs are the drugs that we just cannot increase the drug doses because you would reach like a certain threshold, which will be problematic for the patient. So these are what we're mostly thinking of, oncology drugs. And the third target are drugs that already passed the BBB but have a side effect as when you increase the doses. So we're thinking of drugs, for example, for such as levodopa for Parkinson's disease. The good news I was telling, so we already had some very good study results for the preclinical for the drug delivery. And our next target, so it's for metachromatic leukodystrophy, so MLD. And we're hoping to start this in next January with some mice model and some non-human primates. And hopefully we could be first in patient in around 16 to 24 months. We will use a vector. This will be for gene therapy using the vector and our technology to disrupt the BBB and hopefully reach the CNS to target this. And our final one is glioblastoma. We also had some good results, albeit dispersed in vitro results to target this deadly tumor, as unfortunately, all patients end up passing due to GBM. So we want to see if we are using our technology, we can disrupt especially the invasiveness of the tumor. So that's mostly it. I'm here with Gabriel on the phone, so on the call. So if you have any type of questions on what we're doing or how we're doing it, I'm more than happy to answer them.
Yeah, my question is, thank you for presenting all that. My question is, have you guys thought about neurodevelopmental applications? And I guess in terms of not only children, but trying to see if this technology, whether paired with drugs or not, could have impacts on neurodevelopment, or is that something you don't think is possible, or you're just not, is that just not a focus for you commercially?
I'm guessing not at this stage, but I'm Gabriel, I don't know if you want to.
Yeah, I wanted to add something, but go on and I can complement.
No, go for it.
So, yeah, we're thinking about neurodevelopmental disorders. Actually, this disease that we are hoping to start a clinical trial in the next year is a neurodevelopmental disorder. It's caused by a mutation in some enzymes, and then you get you cannot process some fats, and this process get into the brain and cause a lot of mental disabilities, motor symptoms, sensorial symptoms. So here the idea is that we know that there is a gene that is not working correctly, and with gene therapy we can change the activity of this gene. However, it's not that easy. Because obviously, as you may know, you're experts in all these mutation disorders or disorders that are related to genetics. We have this mutation in all the cells, not just one in one cells. So we need to turn off this gene, and it's actually very difficult to get these viral vectors, this gene therapy inside the brain. So our device, what we hope that will do for these people is to actually improve the delivery of this drug. So for neurodevelopmental disorders, I think that timing is very, very important. And when you do it earlier, you have a better prognosis for the patient, definitely. And in this case, in this specific disorder that we are going to start, there are actually very, very good results in patients. Once they have the drug, they actually increase their life expectancy by 15 years, more or less. So I think there is a lot of potential in neurodevelopmental disorders.
So it sounds like it's more like this technology is almost like a delivery technology to the brain, as opposed to the direct stimulation would itself be therapeutic.
Exactly. Yeah. For this example, it is. We have other disorders in which we directly stimulate, for example, certain areas of the brain or certain frequencies of the brain. I think the best example is Alzheimer's disease, in which there is known that there is an aberrant oscillations in the gamma range of these brains. So what we do is we try to synchronize the firing of the neurons to the correct frequency, like some pacemaker for the heart, but for the brain. So we're trying to do that. Our technology is more like a platform in the sense that we can modify the frequency, intensity. We can modify a lot of parameters, and by modifying these parameters, we obviously change the physics of the device. But this can attack or target several mechanisms of action or several biological processes.
Great. Anyone else have any questions here? We're almost up at the halfway point. I don't want to monopolize the question time. Anyone else, whether scientist or family, if you have a question for Actipulse, now is your chance.
I'll just shout out then. I'm not sure I followed completely. So with the blood-brain barrier permeability, does the electrical impulse sort of increase the porosity? Is it to allow the drug to go through? Is that what it's doing? Is that what you're sort of proposing?
More or less is what is happening. The only difference is that we don't use electric fields, but magnetic fields. So our main working hypothesis is that by using these high frequency magnetic fields, we are changing or we are making the magnetic particles that we have in our blood, because hemoglobin, when it's deoxygenized, it is kind of magnetic. So by applying these very high frequency magnetic fields, we are moving, or we're creating some kind of kinetic energy in the blood, and this creates a mechanical disruption of the blood-brain barrier. So it's obviously what we're testing in animals is that this is reversible. We have tested that it works, but obviously we want it to be reversible because the blood-brain barrier is there because we need it. It's not like I just open it up and that's it. No, we actually need it. So we need to find the equilibrium in opening the barrier, but closing it at will. You know, that's the thing. Because if we can do it at will, we can increase the delivery of the drug in 2 times, 3 times, and that will help a lot of patients.
Absolutely, it's exciting stuff, but also, you know, slightly scary, because as you say,
sounds scary. Yeah.
If there was a mishap and you couldn't close it, you know, that's certainly
exactly. That's why we need to do a lot of research in mice, and then we are going into non-human primates, and after that we are going into patients with this kind of disorders.
Exciting.
I have a question. Oh, sorry.
No, carry on. Yeah.
Okay, so what functions exactly you feel like it's working on? Is it motor functions? Is it like brain function?
It's interesting. Let's talk about transcranial magnetic stimulation as a whole. You know, that this BBB device that is very specific, but transcranial magnetic as a whole, it works by creating this magnetic field that is emitted from a coil, and this magnetic field, as it changes rapidly, it creates electric stimulation inside the brain. So we, in the end, create some kind of electric field, but by inducing it with a coil. So, for example, when we apply this kind of magnetic fields over the motor cortex, we can have changes over the motor cortex. So it's a little bit difficult to say that I will improve the motor symptoms or it will improve the cognitive symptoms. It depends on the area of the brain that we are targeting. It depends, obviously, on the patient. It depends a lot. For example, there are a lot of trials analyzing cognitive rehabilitation in patients that have suffered a stroke. And it works, but the best prognosis factor here is how big the stroke it was, you know, because obviously it depends on how the brain and the plasticity or the internal plasticity. It's not as easy just to say that it will help all the patients. It will depend a lot on the specific of the frequency that we're applying, the brain that we're applying it, and the intensity, the region of the brain. And it depends a lot. There are a lot of variables there.
Okay.
Thanks for your question. Any other questions here before we shift gears to the roadmap review?
Yeah, I had a question for Gabriel. So, so the strength of the magnetic field, I just took a look at your 3 papers on PubMed. The strength of the magnetic field, I think you indicated somewhere, was like 1 Tesla. right?
No,
So
it's one hundredth part of a Tesla. We are using low intensity magnetic pulses.
so well, it's in your abstract, so that's why I said 1 Tesla. But how does that compare to when you're in an MRI scanner? Because those
The
are
main difference, yeah, the
right?
main difference with an MRI scanner are magnitude and frequency. What happens when we're in an MRI scanner? We have a constant magnetic field, and when we're applying TMS, the magnetic field is not constant; it goes up and down. We apply it in pulses, so that's the main difference. Then the intensity for we to create an image out of the excitation of protons inside the brain, that's the MRI, we need to have a very, very strong magnetic field. Actually, if it's stronger, it's better because we can align better the atoms of protons. With TMS, we don't really need the intensity. Yeah, we need it because we need to activate the neurons,
Gabriel.
but we can have biological effects even at very low intensity.
Gabriel,
So-
can you resume because they are on the they have
Yeah,
swamp
sorry,
time. I
sorry.
know you could speak for hours about this. So.
Yeah, sorry. Just resuming, the intensity is different. Yeah, we use lower intensity magnetic pulses that have biological effect, and the frequency we are applied as pulses, not as a constant magnetic field.
Okay, thank you.
Awesome. I think with that, just give a little bit of a gratitude and round of applause for Adrian and Gabriel for joining us today. Thank you guys so much. We're going to shift gears here and talk about the roadmap. You guys can hang out if you want, or you can go on with your days and keep working on your product.
Yeah, and I just wanted to say thank you so much for like sharing this with us. I really appreciate
Oh, thank you. Really, really excited about what you're doing. I think it's always great for us to learn about what is happening in families, what are the real necessities of parents. I think it's very, very important for us.
Well,
Yeah.
we feel the same. And once we start to flesh out the segment 5 of the roadmap, we'd love to come back to you for some technical review and other commentary. So really appreciate you guys joining, and thanks again.
Thank you.
Thank
Stay
you very
safe.
much.
Thanks, Ethan.
Thanks, guys.
Bye.
Yeah, bye.
All right, so we'll shift gears here now, and we'll talk about, we'll kind of check in on the halfway point of our 2-year plan. Let me just get my screen shared again. Okay. So as a reminder, a quick refresher here, we originally conceived of 3 segments of this roadmap and very loosely kind of analogized to segments of a chromosome as sort of this guiding conceptual architecture for how we think about this roadmap. So segment 1, we call drug repurposing. That obviously involves finding uses for old drugs. The reason why we do that is because that's the fastest and cheapest path to finding something that you can take from the lab into patients and see potential benefit. So I'll give you an update on where things stand on repurposing after at this midway point and what's beyond. Second segment we called neurological disease modeling, and I would say that's where this past year was really a bonanza of just amazing and unexpected results. And I think real it ended up being a real legitimizing proof that using brain organoids, for example, and neuroprogenitor cells and neurons going that full stack was actually a good idea, because we see convergences between those models. And so that was ended up being, I think, a really good bet that has paid off. Segment 3 is chromosome therapies. This is our moonshot idea, applying the concept of a gene therapy where you might fix a single broken gene, but expanding that to fixing an entire broken chromosome. Obviously, this is going to be a multi-step process, and we're just getting the foundation laid, and we'll talk about the progress that's been made so far. I'll pass the mic back to Bina to cover segment 4, a health data platform, or as we kind of affectionately call it, the InHero studies. And then, of course, segment 5, we just alluded to, is not fleshed out yet, but will be added to the roadmap in moving forward in the kind of refresh that I'll be doing at the end of the year of the document. So this is a figure that Whitney put together, trying to summarize the interactions between these different segments. It's maybe a little bit confusing to watch, to see the flow, because there's so much integration with all these tracks or these segments. That was intentional, that they would all be feeding into each other. But we will also refresh this diagram to include the segment 5, but also to try to flesh out some of these relationships here and be a little bit more specific now that we've actually have a foundation of results that we can build on. Okay, so segment one, I will just sort of do a recap here. I'm not going to really refresh. I'm sorry, I'm not going to really recount all of the various data that kind of backs all of this up. Some of the data we put out on our on the Chromo AP Substack. So you can either you read that or you can follow up. So I'm not going to refresh all of that. I just wanted to do a high level summary here of what sort of happened with each of these projects and what happens next. So on the drug repurposing side, we started out with the lowest barrier to entry, which was doing an AI-based approach. Really, probably it's a little bit exaggerating to call that AI necessarily, but it was let's say a bioinformatic type approach where this was spearheaded by UAB and Matt Might's group, where they essentially could ingest all the abstracts from PubMed and build a knowledge graph and start to be able to say things like gene A inhibits gene B and drug C, activates gene A, or build these kinds of knowledge graphs where you can start to make some predictions. In this case, what drug might, for example, be beneficial for a particular genetic disease. Now, of course, AP is not just a monogenic disease, so this was always a kind of a stacked deck here in terms of what this knowledge graph could really produce for AP. And there were a bunch of predictions. If you recall, we had a roundtable last year where we had Alexandra Foxina, who was the representative from UAB, who gave a talk and described what they found. And the bottom line was that nothing really emerged from that exercise that was practical, but I think it still was a good thing to do. And I said, probably is a little bit of a stretch to call that AI. This was all done basically before ChatGPT and LLMs blew up on the scene. So you can imagine revisiting this AI-based repurposing approach, but I think it's still always going to be fraught, given that this is not just a monogenic disease where we go after a single target or single pathway. So I don't know if anyone else has any comments on what we would do on the AI-based side of repurposing, but definitely open to suggestions. But I think at this point, we kind of examine the low-hanging fruit, and there really wasn't anything there, but doesn't mean we can't revisit AI repurposing in the future. If anyone has any comments, feel free to jump in.
Actually, I just
go
had
ahead.
a question. I just had a question related to that, which is the drugs that the 8P heroes currently might be on. Is there a way of, I don't know, is there a way of sharing that information in a appropriate manner, in a protected manner, so that we could have an idea of what kind of drugs, for example, they're taking for epilepsy or for their other comorbidities?
Definitely. I mean, maybe Bina, you want to address this in the segment 4 section, or maybe you want to address it right now.
Yeah, so that's a really good question. And I think that Katie and I are kind of thinking through the next step of surveys that we push out. We do have like AEDs, like the anti-epileptic ones that we ask about for those that have seizures. But when it comes to like the entire set, I mean, frankly, my daughter has about 15 to 20 supplements that she takes, and we rotate them and we kind of run our own little mini household trials, right? And so that is not documented in our database. And so we do think that's kind of the next step as we head into designing, kind of a pilot repurposing of could be just nutraceuticals. A powerful antioxidant, for example, or something like that. So, it's a really important question, but absolutely. I'll also talk about, how whatever we do in this community, we would remove any patient identifying information, but we do want to, as long as families opt in to sharing the data with researchers, that would be available to all of you.
I mean, I was even asking just about prescription drugs, not even supplements, because I could imagine that would be very hard to keep track of. But even if there was just some information about what prescription drugs have been applied for various things that may co-occur with 8P, that'd be very helpful to know. Thank you.
If you ever visit the home of any family with a kid with a rare disease, you're going to find this massive pharmacopoeia of supplements and other things. But I think, yes, getting all that squared away as kind of a baselines, especially if we're going to try new interventions, I think that's exactly what segment 4 and the data portal and sort of everything around, establishing not just genotype phenotype relationships, but also these chemotype phenotype relationships. So yeah, this is great for pointing that out. Okay.
And thank you for the feedback, because that lets Bina and I know that this is a track we should grow, that you're interested in. So thank you.
I was also just going to say it's important to also for us to coordinate how long, people are on these drugs, because kids come on and off drugs, and then, what's the effect, right? And what else might have been happening at that time, right? So like certainly with Madison, much to my shock, we found gluten is actually a huge issue for her. It's the difference between sleep and no sleep. Gluten, no sleep. But I was just wanted to say, Ethan, that I completely agree with you about the AI, and I think like revisit this in 5 years and it'll be a whole new landscape, right? Like so.
Maybe even a year. I mean, things are moving super fast. Yeah,
Absolutely.
It's
Yeah,
just moving
I hedge bets.
Yeah, exactly. So rare base, let me just quickly summarize. So the approach here was to generate glutamatergic excitatory neurons and to perform a drug repurposing screen based on RNA-seq, basically changing gene expression levels. And again, this was almost sort of like a trap, because this was really designed for a monogenic gene. And essentially AP is like a, dozens, maybe hundreds of sort of monogenic diseases essentially rolled into one. So what we ended up with rare base was we could look at individual genes that were downregulated or that were in the deleted segment, for example, and ask, were there any known drugs that could increase the expression of one of those haploinsufficient genes? And the answer is yes, you could find, rare base could find examples of drugs that could do that. But looking at those lists, it wasn't really clear, A, that there was a cohesive mechanism among those predicted hits, and B, the particular haploinsufficient gene you chose wasn't necessarily going to be the driver, right? And we can go back to the concept of driver genes in the first place. But I think just like with the AI, this was a stacked problem, because this is not a monogenic case. Even if you found examples of compounds that could, remedy a particular genetic haploinsufficiency, what about all the other genes? Maybe some of those don't matter, right? You'd argue only a few are drivers, but we were still caught in that circle level. We don't know what a driver is. So which one of these haploinsufficient deleted genes would we prioritize, and why would we assume that any one of them, if we increase the expression of the mRNA, first of all, how do we know that increasing the expression of mRNA would increase the expression of the protein? That was always sort of a question with this approach. But even then, if you could be guaranteed that the protein levels went up, how do you know that would matter? And maybe it would have mattered if you did it early off in development, but maybe not late. And so there are all these questions that just didn't lead to an obvious next step. But again, I think this could be something that could definitely be revisited, especially as there are other approaches we can take to repurposing, as I'll get to with fibroblasts, where maybe eventually we can connect back either to the rare bases data set or even explore another RNA-seq based platform approach, because there are other competing RNA-seq platform approaches out there as well. And then I think the area where we have this most solid footing in terms of being able to make next steps in the second year of the plan is with fibroblast-based repurposing. So Martin from Nicoletta's lab, spent a lot of time over the past year phenotyping and characterizing a panel of 8p fibroblasts versus controls, and was able to identify a number of cell-based morphological phenotypes. We call that cell painting. This collection of assays where you're looking at differences in cell staining of different compartments and comparing disease cells versus wild type cells. So we're at the position now where we're trying to transfer the project from the academic setting, where we did this assay development and proof of concept, and moving it into more of an industrialized screening setting. And that's why we've started a conversation with Charles River. I've worked with them. Pilar has worked with other clients that have benefited from Charles River's high throughput capabilities. They're really kind of unrivaled in terms of their speed. They're obviously, comes at a cost, of course, but they are kind of consummate professionals at the screening part of this. So we're hopefully going to be booting up a repurposing fibroblast-based repurposing screen with Charles River, based on all the great foundational work that Nicoletta's lab had done. And if you want to know more about that work, you can look at the Substack from earlier this summer, Skin Deep, where we kind of talk about and summarize all the different phenotyping differences that Nicoletta was able to see comparing 8p fibros to control fibros. So we're really excited to take those fibroblasts and take them to this next stage where we can run a screen and start to actually identify novel mechanisms, novel hits. So that's what we have to look forward to, I think, in the second half of the year for drug repurposing. And if there's any other comments, feel free to jump in. Otherwise, I'm going to, in the interest of time, shift to segment 2. So this is, as I said before, this was, I think, the segment that has been the most bountiful in terms of unexpected results and results, period. And the major partners here we've been working with are at UC San Diego, Harui, working on neuroprogenitor cells, and not just, but starting there. Obviously, Allison working on the brain organoids, and then Rarebase as well, having created the NGN2 positive excitatory neurons. So we have this sort of full stack of neuro-focused disease models, and each of them yielded something interesting. And I think the major discovery that we found was the overexpression of relin, which I'll get to kind of in the summation slide. But so far, I think the neuro-focused disease modeling strategy has paid off in terms of going from the kind of two-dimensional pure neuron population to the neuroprogenitor cells that are going to be the ones that are causing these neurodevelopmental phenotypes, and then the brain organoids, where we're trying to capture as much of the complexity as we can outside of the actual human body. So I won't recap all the results here. Again, I just want to highlight that relin was a discovery that connects all three of these model systems, and specifically relin overexpression was observed in all three of these model systems. So again, more confirmation that doing this full stack approach was a good idea. I'll pause for any comments or questions on the neurofocused disease modeling. Obviously, we haven't done any other tissues yet. Very, very focused on neurological symptoms and neurological and neurodevelopment. But one can imagine also at some point in the future thinking about other types of tissues, but that's not something we're really contemplating at this point. So if anyone has any comments on the neurofocused models, go ahead and chime in. Otherwise, I will move on to segment 3. So here, I think I just wanted to, again, summarize at a high level what the strategy is. So working with Shelter's group, Jason's on the call, and we're trying to take what I think we're calling a top-down approach, which is starting with 8p cells and then trying to get on the other side euploid cells, and doing all kinds of tricks in between that are kind of captured by this black arrow here. But the idea is that you're almost letting the system itself find the answer. And then there have the complementary bottom-up approach. Stefan's lab at UConn is building these CRISPR-I and CRISPR-A systems into a euploid line. And so while we can start to, from the bottom up, identify particular drivers or particular segments and start to assess what are their effects on phenotypes. And ideally, there's going to be a convergence in the middle here between these two approaches. And those will be the tools and the reagents we have where we could one day start to try out these experimental chromosome therapies. But we first have to establish the cellular systems and also establish the pathways to euploidy. So those approaches are underway. I think Jason presented recently the results that Sophia has produced so far that are quite encouraging. And we have some potential clones that look like they're on their path to euploidizing. And Stefan's group has done a great job of creating these CRISPR-I and CRISPR-A reagents that are going to be useful for so many different downstream purposes. And then segment 4, I'll pass the mic to Bina here to cover the progress we've made in HERO studies.
Hey, thanks everyone. I know there's only 10 minutes left, so I guess I'll just sum this up in 1 minute. But the most important thing is the 8p family community that gets really involved, because we can't help answer any questions that our amazing scientists have, or any other partners, without knowing exactly what's happening to the 8p HERO over time, right? What has happened in their past, what interventions they're using, like, for example, some of the drugs that could be helping them, and then, what they look like today. So this is so critical, and there's a lot of different ways to get involved. Obviously, my HERO initiative is just kind of an easy way to explain that. You can get involved by the 8p registry. We have a growing biorepository, and we have an amazing group of families that are actively continuing to enroll in donating their biospecimens. And then one of the bigger things that we have is we obviously have had this CNV portal done by Dennis Lahl when we launched the commission on CNVs. And that's still current and active. And he is now at Texas Children's Baylor in Houston, and he's really very engaged in growing this and adding more phenotypes and information. We also had Illumina present on advanced genome sequencing data across all of our sample sets. And there's a part 2 meeting this week. So if any of you want to participate in that, please let me know. And then our clinic for sure. And by the way, I'm speaking a little softly because Ethan and I are technically in the same space and I can hear my own echo, and vice versa. So the neurogenetics clinic is, our first site. We hope to grow and expand this. And that's, so important because Katie's really taking the lead as our full-time patient engagement manager for 8P and trying to help them draft care guidelines, care recommendations. They've seen over 30 8P heroes in the clinic across 10 different providers. So that's a really important data set that we have. And then overall, what we've built and we will launch very soon is our insights portal. Super excited about it. It is so that researchers can access phenotypic data. They can filter their queries based on different symptoms across many different organ systems, based on different genotype buckets, like whether they have a deletion only or duplication, they have additional variants. And so we're kind of building that right now to take in all these different disparate data sets so that the administrators, even myself, can make really important decisions. We can start generating real-time insights back to our community. So we're really excited about that. And that you don't actually see on this slide, but we'll probably do another roundtable just dedicated on that.
Great. Okay, so to summarize then, looking ahead to the next year, oh, we got another chat. Oh, great. Thanks, Faye. Okay, looking ahead to next year, as I said, we are pivoting from the assay development and proof of concept that there are differences between 8P fibros and wild-type fibros with respect to these cell painting phenotypes. So we're kind of full steam ahead on taking that to the screening step with Charles River. And we're actually in touch with Ann Carpenter's group at the Broad. That's the lab that develops cell painting. I've been working with them on another project, a monogenic disease where cell painting on fibroblasts has successfully identified some really interesting candidates. So that for me was a good proof of concept. So I think we're going into cell painting and fibroblasts with sort of, I think, the best team and best advice we have. But obviously there's risk whenever you do anything, and I don't necessarily want to relitigate all the questions about fibroblasts and their utility as a model and so forth. I think, we can have lots of discussions about that probably to the end of time, but I think our feeling is that this is a good idea and that at some point this could also be a template for doing a similar screen in neuroprogenitor cells. And in some ways, the fibroblast could be kind of practice and setting the stage for the next level of repurposing, which could involve a more neuronal specific cell. So I think the major discovery that we made over the last year was that relin appears to be an 8P driver gene. I'm not sure if it meets that definition. I don't know what technically a definition of a driver gene would be, but maybe this is something we could talk about a little bit here. But I think we definitely want to follow this thread, pull on this thread some more and see where it leads, given the prominence of relin in neuroscience and how there has been such a focus on the loss of function phenotypes. And what we're proposing here is a gain of function phenotype because this is all overexpression of relin. So maybe I'll pause here for any thoughts on what how excited to be about relin, how much to really double down. Obviously, there probably will be other quote unquote driver genes that will capture our attention, but maybe a few minutes here of discussion about relin and that result and how best to exploit that. Maybe one of our scientists can jump in if they have any thoughts. If not, it's okay. I think this is something that we're going to be double clicking on for sure. And this is be an ongoing conversation. So anyone who wants to, maybe we'll have to start a relin subgroup or something. But if there are no comments now, then we can revisit this. But obviously that's on our radar big time.
I think Nicoletta has her hand up.
Sorry, I'm not able to see who has their hands up. But Nicoletta, go for it.
Okay, so I'm not really expert in relin, but I think if it came through consistently in all these models, perhaps it's worth looking at it. And I guess the easiest way would be downregulation of it in some relevant models and see what happens.
Right. And I think this opens up a big question as to how would you do that? Would you do that at the ASO level? Would you do that siRNA level? Would you do it at the mRNA level? Would you do it at the protein level? This is a secreted protein, right? And there's lots of complications and nuances to its signal transduction pathway. But yeah, I think probably the simplest experiment would be to intercept it at the mRNA level somehow. Seems
the
like
easiest, yeah.
Yeah.
Yeah. Sorry, Nicoletta.
Go for it. No, hurry, go for it.
It's actually not as simple as just a gene upregulation. So renin is a marker of something bigger, right? So we're talking about, a subset of neuronal types, which mostly is known as like Cajal-Rhesus neurons. And these are play different roles at different times of development. So early on, they're involved in basically structuring the hippocampus. So they can be involved in learning and memory dysfunctions, but also later on, they're involved in neurogenesis and proliferation of that region, also cortical layering. So knocking it out is just going to drive everything towards the knockout system, right? So we'll have models that basically recapitulate the knockout mouse. But we believe that there is a more complex sort of regulation that's leading cells, neuroprogenitor cells, to shift or differentiate more into these renin positive cells. So when we look at the single cell RNA sequencing data from Allison's group, you see that that population is expanding. So it's not just that the gene itself is upregulated, it's just as a population, neuroprogenitor cells seem to prefer differentiating that way. And we don't understand why. So I think right now we're trying to figure that out. But it has significant impact on learning and memory, brain development. So that's why it's really, really exciting to try to figure out the role of these neuronal subtypes and how they come to be and where they go. I think all of the studies that have ever been done on renin and Cajal-Rhesus neurons has always been on mouse tissue. So now that we can recapitulate this utilizing stem cells and organoid models, that's something that I guess we're trying to figure out.
Awesome. Thank you for that.
Thank you. Sounds great. Thank you.
Can I just ask, is it associated with any other disease that known of?
Renin?
So
yeah. I asked the most obvious question ever. Sorry.
No. Go ahead.
Yeah, so there have been several disorders where there's disruption in that gene, and upstream genes or downstream genes that are associated with that are involved in intellectual disabilities.
Okay. It's already associated with ID. Okay. Great. Thank you.
But
it's
associated with schizophrenia. I mean, bipolar disorder, autism.
Okay.
And there are protective mutations in renin that seem to protect against Alzheimer's. So it's sort of, it's everywhere and
an obvious go-to.
Yes, so that's why it was so kind of exciting to see it. But then, as Harui said, there's then many complications that ensue, given how entangled it is in all these processes throughout life, in neurodevelopment and also in neurodegeneration later in life. So it's not something you can just turn off as a switch.
No, but lots of other sort of systems to look at and compare with, etc., which is helpful.
And I think that feeds into the third point, which is that starting with a comparison, just sort of a naive comparison of fibroblast transcriptional data, looking at trisomy 21 fibroblasts and 8p fibroblasts, we could see a convergence where about half of the transcripts that were up in trisomy 21 fibros were also up in AP, and same thing for the downregulated transcripts. So this convergence seems to us to be pretty exciting, and is sort of, I think, a nice complement to this thought of individual driver genes. And so I think we'll definitely be exploring these convergences more and more in the year ahead. And then I also hope that in 2024, we can not just simply be in the lab, but we can start to move these discoveries from the lab into single patient studies. And I frame it as 1 to N, because it's not just a single patient and that's it. It's that it's a pioneer, and then there's a second pioneer and a third pioneer, and how high you get, just what that N is depends on just how successful the intervention is. And it doesn't just apply to a repurposed drug. You can, we've learned about augmentative devices, some of which could be considered electroceuticals. And we've heard about nutraceuticals as well. So the bottom line is we need to sort of take these in a thoughtful and data-driven way into the clinic and really start to understand, do these create benefit? So I think we have much to look forward to in 2024. I know we're at the bottom of the hour here. So if there are any other thoughts or feedback, please, stage is yours here. Otherwise, we will wrap up.
I mean, I had a question. I think that, this required like a big workshop where all of our researchers and maybe some of our advisors who are not actively necessarily studying 8P could join. So would that be something that many of you that join these roundtables would be open to, and really putting our like roll up the sleeves workshop, and then kind of heading into, you know, our scientific meeting, which is planned for the end of July. So if we haven't sent you those dates, we will, but that would be like our family and science conference. But we'd like to kind of do that in the next month. Is that, you know, thumbs up if we can send you some date options for that? I need to see more than Ethan.
Come on, folks. There we go.
Is
All right,
this
we'll
like
follow up.
meeting in the next month to just chat about everything?
Yeah, I think
guess?
a more intensive brainstorm here, and maybe going through some like the discussion we were just having about Reelin, for example, and maybe having that be a little bit longer discussion and think about that a little bit more thoughtfully. Okay.
Thanks.
All right, we have a quorum.
All right. Thanks. Yeah, I think there's a lot of details we need to dig into, and we need to really understand, you know, like where should our next set of funding go towards, right? And so we want to just have, it's going to be hard because it's like remove your lab out of this conversation maybe, and just say like, here are all the great things that we really need to understand. Here are the big questions we don't know about 8P biology, and then tackle it, and then kind of say, okay, here, each of your labs can actually answer parts of this collectively. So I think it'll be a really important exercise for all of us to do. It's probably not typical in the way rare disease organizations or patient advocacy groups are, but like, look, we're not typical. We've proven that time and time again. So we're in this together. We really mean it. So, you know, putting together like a research strategic plan requires all of our input.
Here, here. Thank you, everybody, for attending. And yeah, appreciate everyone's feedback and constant engagement. This roundtable has been, I think, a wonderful gathering for us and place for brainstorming and sharing. So again, it doesn't work if you don't show up. So really appreciate you doing that month in, month out.
Thank you. Bye.
Bye, everybody.